And our last speaker is Felix Baerenz. Felix is a lab head in the R&D department for integrated drug discovery at Sanofi. And Felix lab is actually the lab with the most Surfer SE publications and early adopter of the Surfer SE technology, I would say. So thanks a lot, Felix, that you're here with us.
Let's get started. So I'm Felix from Sanofi, here located here in Frankfurt. Greetings to Munich. Let's start. So today I'm going to present you a few assays which we recently established, also using the Surfer SE platform. The title of my talk is actually the development of high-throughput SLC assays applying SSM-based electrophysiology, as well as, this is currently missing here in the name of my talk, acoustic droplet ejection mass spectrometry. First, anyway, I will give you a brief introduction again into SSM-based electrophysiology in general. I don't think I need to talk a lot about it because I think you have heard a lot of different other talks on this topic. But I will also mention a second technology which we are applying, which is called acoustic droplet ejection mass spectrometry. Then the first assay I want to present you today is a solute carrier 1 family assay, where we applied both technologies, and this story was recently published, so you can check NCBI if you're interested. And in the last part of my talk today, I will also present you recent work from our lab. It's an SLC 6 family assay applying the same technology. And last but not least, I will give you a brief summary and an outlook. So the first technology which we are using in a high content fashion, primarily as a secondary or tertiary assay in a drug discovery screening cascade, is the SSM-based electrophysiology, and we are applying heavily the Surfer SE system from Nanion. I will not go here into details. I think you are super familiar with the technology. I just want to mention that you can easily measure electrogenic transporters, especially solute carriers, using this technology, because you can, by the addition of your substrate and the imported sodium ion, for example, you can easily generate a current which you can measure, and you can do this in a high content fashion, because the system allows up to 96 wells, and you can measure in parallel. I mean, the department I'm working in is early drug discovery, and we are using the Surfer SE platform, as I mentioned, as a tertiary assay platform in a high content fashion. So we are measuring different compounds, different concentrations, different targets in parallel. And the Surfer SE, I think, is an ideal system when you want to do SSM-based electrophysiology in a high content fashion. And we recently published different protocols which you can use to apply a SSM-based electrophysiology in a high content fashion. If you are interested, please check out our current protocols publication. The second technology which we are using as a so-called entry or primary assay is a rather new technology, I would say. It's called acoustic droplet ejection mass spectrometry. It was published and established by a mass spectrometry company, SciEx, I think 5 years ago. It combines acoustic droplet ejection technology with standard mass spectrometry. And what is basically the reason why we are using it? Because it allows a very high throughput in the absence of any separation of your sample, and you can measure a complete 384 well plate, for example, in around 5 to 15 minutes, which is rather impossible by doing classical LC or GC-MS. even the SPE mass spectrometry system from Agilent, I would say, is 4 to 5 times slower in throughput. How does it basically work? You have a 384 well plate with your sample in, then an acoustic transducer generates an acoustic signal which leads to the evaporation of small droplets in the size of 2.5 nanoliters. And these droplets from your sample are directly injected in a so-called open port interface, and a carrier solvent, which is basically sent into this capillary, transfers your sample directly into the source of your MS. So you can see here, by using small droplets in a rather huge carrier solvent flow, heavily dilutes your samples, avoiding matrix effects, and allows a very high throughput, because the complete separation of your sample, what you normally have using SPE or LC separation, is completely absent, and you generate a very high throughput. Another advantage when you're talking about transporters in general or solute carriers, you can easily use this technology in a cellular setting to measure the import of different substrates of a well-established or unknown SLC to check the import of your substrate into the cell. And this is what we have been doing. Just a brief introduction to solute carrier proteins. You will find more than 400 in the human genome. All these members are divided into 66 different families, and they are pretty relevant because they maintain cellular homeostasis. They are also relevant in different diseases, including cancer, neurological disorders, diabetes as well. And interestingly, there are only 22 approved drugs available. And there are so many of them with so many disease-relevant functions that it is very important to establish assays to measure the transport of these SLCs to be able to start a drug discovery campaign. And a few years ago, you might have heard of, there was an IMI consortium called Resolute. It was an EU funded project, including 13 partners from science and industry. And what was actually the main goal of this IMI consortium? It was to establish assays for deorphanization of SLCs, and also to generate knowledge, to generate assays, and to support the research community in general through freely accessible databases. So all the assays which we established and all these tools the entire project team generated are now available online just for free. Yeah. So whenever you are interested in a novel, unknown, or interesting SLC, just go on the Resolute page and check out the different tools which are currently available. And Sanofi was one of the 13, was one partner, and we established several SLC assays for potential drug discovery campaign. And the first story I want to present today were two assays which we established for SLC1 family members, the so-called EAAT family. And if you are interested, go to NCBI, you will find this publication. We published the entire story earlier this year in Frontiers in Pharmacology. So if you're interested in these assays, please check out our current publication. Just a brief background on EAAT family. EAAT SLCs are glutamate transporters. They move glutamate across the membrane of excitatory neurotransmitting cells. They are heavily expressed in the brain, especially in the synaptic cleft, and they regulate the glutamate reuptake into glia cells and neurons, so very important transporters. Interestingly, when you check for tool compounds, you only find inhibitors of these transporters, but when you're talking about potential drugs, you might require or need activators of those SLCs. But interestingly, if you check literature, only a few inhibitors are known. And the first thing we basically did was to establish an acoustic droplet ejection mass spectrometry assay to be able to screen full deck libraries consisting of millions of compounds. And to establish such an assay, the first thing we are doing, we were using HEC293 wild type, as well as HEC293 cells overexpressing EAT1, 2, and 3, and checked for glutamic acid uptake into the cells. And as you can see here very nicely, the wild type cells only showed basal import of glutamate, and cells expressing the EAT, especially EAT1 and 2, heavily imported glutamic acid in a cell count dependent manner. And we also tested a sodium dependency. So EATs are electrogenic SLCs, which generate a sodium gradient across the membrane. And so we checked sodium dependency, and we could show also in this assay already that the transport of glutamic acid is definitely sodium dependent. Then we also checked the substrate incubation time, because when you do a high throughput assay, everything needs to be fast. So we also checked whether we could use this assay by incubating the cells with the substrate for only 30 minutes. And after 30 minutes, we already could observe a nice uptake of glutamic acid, especially for EAT1 and 2. You might ask me later, why don't you see a very good import of EAT3? EAT3 expression in those cells was decreased, so it was not so nicely expressed. And also the import kinetics seem to be different to EAT1 and 2. And also the structural features of EAT3 might also be responsible for the decreased transport rate, but it is not actually the topic of today's talk. Last but not least, we also checked DMSO tolerance, because all the compounds we normally screen are dissolved in DMSO. And yes, you can see here quite nicely that the transport itself is not heavily affected by the presence of DMSO. And then in a very last experiment here, we also determined the Km value for glutamic acid using the 3 different EATs. Now, it seemed to be more interesting to the audience here today. We also established a surfer SE assay here. And the first thing we did, so we prepared membranes from HEC293 cells expressing EAT1, 2, and 3. In the first experiment, we checked currents at different membrane concentrations, and you can see here already nicely that we can observe a saturation, and we observe a membrane concentration dependent increase of currents in the presence of glutamic acid and sodium. Then we also again checked sodium dependency, and as seen in the acoustic droplet ejection mass spectrometry assay, also in the surfer assay, we could see a sodium-dependent increase in EAT currents, which is very much in line with literature. We also determined the Km value using surfer. And if you check out our publication, you will see that there is for sure a significant difference in Km value comparing the biophysical surfer method with the cell-based ADE assay. And of course, we checked DMSO tolerance. In a very last experiment, we screened at single concentration at 10 micromolar, a small Sanofi internal compound library into replicates. And you can directly see here that compounds fulfilling our hit criteria show a very good correlation in terms of activity comparing replicate 1 and 2. We also had a very robust and stable signal to blank comparing two replicates, and also the set prime value, which is one of our QC criterion, stayed constant, higher than 0.5. We further validated initial hits in dose response and also compared the IC50 value using ADE-MS with SSM-based electrophysiology, and you can directly see here that some of the compounds we identified as potential primary hits have a very similar IC50 value, so inhibition value in both assay systems, and on the other hand, there are also a few compounds which have a significant different IC50 value comparing the two different technologies. But the reasons behind that, I will not go into the details right now, but I will now switch to some unpublished work, especially work we have currently done for SLC6A15. SLC6A15 belongs to solute carrier family 6, consisting of 4 groups, and SLC6A15 belongs to group number 4, transporting neutral amino acids, especially proline, leucine, isoleucine, and valine. It's localized also in the central nervous system. It's a symporter. It transports sodium together with these neutral amino acids, and it's also an electrogenic transporter, so you can basically apply in principle SSM-based electrophysiology. So we started with assay development on ADE using our mass spectrometry approach. I will not go into all the details illustrated right here. So we first checked different carrier solvents to check we basically checked the system, whether we can establish all the different neutral amino acids. And this here is the illustration for one of the amino acids which we tuned. We also checked how many seconds do we need in between each well, and we found out that already after 1 second we can easily observe nicely separated signals, so we are pretty fast here. We also checked the correlation, so we did a proline titration here and checked dose dependency, and we observed nice dose dependency for proline in matrix and generated an R-square value of higher than 0.9. We also checked a tool combined loratadine and checked cell count, sodium dependency, etc., and generated also values for that using proline as a tool substrate for SLC6A15. And we're also comparing loratadine as a tool compound with cells treated with DMSO. And we also generated here a nice split between wild type or untreated cells with loratadine treated cells. When we're talking about the surfer assay, in the first step, we were comparing wild type HEC cells with HEC cells overexpressing SLC6A15, and also here in C in the presence of a two compound loratadine. And you can see here we observed only basal levels or almost no currents in wild type cells after addition of proline, and in the presence of 6A15 we were able to generate nice and stable currents specific for proline. And in the presence of our tool compound loratadine, illustrated here in C, we were able to significantly decrease the currents. And we later on also tested dose dependency and could show that these currents decrease in a dose-dependent manner, supporting specificity here. Then what we always do here, these are data from my coworker Antje in the lab. We first checked baseline stability. So we did several injections just in assay buffer in the absence of any substrate, so no currents were observed. The complete measurement time here was 5 seconds. By the addition of our substrate proline, we were able to generate a specific current, which is illustrated here in activation number 3. And this is very stable and robust after 3 different activations. And if we now exchange again our buffer infusing proline with our tool compound loratadine, we are also able to significantly decrease the current as shown previously in C. So we finally validated both assays. We also did a few extra experiments on surfer, what I have shown you previously for the other SLC, the EATs. We also checked different membrane fractions, and here you can see that the best signal to blank was generated using a fraction between 9 and 31% in the sucrose gradient. And we also could show a membrane-dependent increase of the currents with a saturation around 6 microgram per surfer sensor. We could prove and show a sodium dependency, and were able to generate a Km value. Then again, we did a high content screening more than 5,000 compounds in total. Around 8,000 compounds of our library were screened using the mass spectrometry approach. We were able to generate 38 primary hits, which is equal to a hit rate of 0.48%, which is quite good. Our hit threshold was 37% inhibition, a very nice correlation between replicate 1 and 2. Also, the signal to blank over these 25 plates tested was quite robust, including set prime value. And last but not least, we also checked cell viability, and we could show here that a few compounds, or many of these compounds we identified in this so-called validation library approach were toxic, which is not unexpected. And the non-toxic compounds were then further validated in parallel in dose response using surfer SE as well as acoustic droplet mass spectrometry. And you can see here that we were able to generate a few hits and included loratadine and tiagabine tool compounds, which were already published as a control. Now I want to briefly summarize everything we have done. So we successfully developed a high-throughput SLC assays for EAT and SLC6 family member using SSM-based electrophysiology applying surfer SE technology. We also developed a specific assay for 6A15 combining surfer and ADE-MS, and we were able to confirm the transport activity for several neutral amino acids which were already published in literature, including proline, leucine, isoleucine, valine. And I want to mention that all these approaches we used here are basically completely label-free, so less artifacts can be observed, and you don't need any radioactive labeling of something, of your substrate, for example, to measure the transport activity. So everything we are doing is basically label-free. And yeah, and potential outlook could be the integration of this transporter platform in our current screening group here in Frankfurt, and to use the surfer as a secondary profiling platform. And to expand, of course, the assay development to additional SLCs, which might be potentially interesting for us. And now I want to acknowledge a few people. I definitely want to acknowledge Antje from my lab team, who's doing all the different assays. She's what was, I think, one of the first people here within Sanofi applying SSM-based electrophysiology together with her former boss, Thomas Licher. And I also want to thank Thomas for his scientific input and critical review of all the data we generated. And last but not least, I want to mention many master students we had here in my group, including Yasmin doing all the EAT work, Catherine doing all the SLC6 work. But in the past, I already had several other students working on this platform, including Kim Lang, Katharina Sattler, and Jennifer Walkentin. Yeah, and for sure, I want to thank the IMI consortium for providing cell lines and membrane batches. And with this, I come to the end. Thank you very much.
Thank you, Felix. Then there are questions for you.
Great talk, thank you. So my question is, as you mentioned yourself, EAT family, pharmacologically speaking, would be more relevant to find activators. And your hit screen primarily showed inhibitors. Do you think that assay is optimized to detect activators of EAT family?
Yeah, sure. Let's maybe go back here. As I mentioned, there are only inhibitors currently published and well known and established for the EAT family. But if you check out here the replicate correlation and the POC values, you can already see that we already also identified several compounds here which show a potential activation of the AATs. And there are a couple of compounds even showing an activation of EATs greater than 50% compared to the untreated transporter. So I think in general, you can also use this platform here to profile and to identify activators of EATs. But you are right, since there are only inhibitors currently published and well known, we were using this as a so-called tool to establish the assay and to validate the assay. But in principle, by using such an approach, you can basically also identify and profile activators. Yeah, that's my current view on that. Oh.
Thank you. There was actually a publication on a EAT2 activator, GT949. I'm not sure if you can share if you ever tried testing it in your lab using surfer technology or even the mass spectrometry.
Yeah, I know this publication, but unfortunately, due to time constrictions, we have never tested it, unfortunately. But I know the publication you are just mentioning. No, we never tested, but yeah, sorry, no, we didn't.
Thank you very much.
I have one question, Felix. You did not go into detail on the differences of the assays in the EAT screen. So you said there were some different results on the surfer versus the mass spec assay.
Yes, you're right.
Is there maybe something you can share or any ideas, speculations about that?
I mean, I know from literature and I also know from our own experience that there are always significant differences sometimes between surfer SSM-based assay and a mass spectrometry import. I can speculate maybe that you are measuring in the SSM-based assay, you're measuring currents, so the currents which are generated during the transport, and in the cell-based assay you are using intact cells, living cells, and you are focusing your measurement on the substrate itself and not the currents behind. So there might be different factors. So you have baseline currents always when you measure the cell import or an import using SSM-based electrophysiology, and on the other hand using the mass spectrometry approach, you always have also background import of your substrate by different other transporters across the membrane. And on surfer, it's more a biophysical cell-free assay with intact membranes, and the cell-based assay is using intact cells, so you might have different transport rates due to the interaction maybe with other cellular proteins which you might lose using the SSM-based assay. And these are all factors, in my opinion, which could affect or impact the inhibition of the transport itself, because in the electrophysiology assay, you might also destroy several substructures which you still have in intact cells. But there might be also other factors. Maybe other people in the audience have different experiences or similar experiences and also can help me explaining this phenomenon.